Hi, I'm Alessio Sweeney from Area Science Park. We are a national public research center and specifically work in the Technological Innovation Research Institute. Modern biology is characterized by the availability of large and complicated datasets, like, for example, DNA sequences. As an example, for humans, DNA sequence of the complete genome is 3.6 billions of these letters. And it's important to develop algorithms to detect, for example, as in this case, the subsequences that code for genes. And by the way, one single mutation in this gene, for example, or another gene could have dramatic consequences, could lead, for example, to a systemic disease, or, for example, a sickle cell disease that changes the shape of red blood cells. And proteins is the starting point where we started to do research. Proteins can be described in this way as sequences, and by comparing also homologous proteins in different organisms. In this way, it's possible to see what is preserved, what is changed, and what is co-evolving during evolution. And on the basis of sophisticated algorithms, it's possible also to predict the three-dimensional structure from a data structure like this. The most important example is AlphaFold, which is the state of the art in this field that we used a lot. And we basically approach the problem of defining families of proteins, which are subsequences that have functional and structural properties. And we address this problem with a technique of unsupervised learning based on the density peak clustering, leading to families that are comparable to the hand-curated datasets. They took 20 years or more to be constructed. And this was a work done in collaboration with Alessandro Lavio in SISSA. And we also compared these families, this structure that we found in the space of proteins against the state-of-the-art language models. In particular, MSA transformers and AlphaFold2, which are really state-of-the-art, a bit different, but both state-of-the-art predictors of contacts, secondary contacts, and then also structure. Machine learning in genomics is a huge field. We are currently interested in a problem to reconstruct the genomes of complex microbial communities, which is a very hard problem because genes are basically mixed when one performs an experiment like this. And there is really very partial information about what to expect. Another problem we are interested in is protein-protein interaction by means of geometric deep learning. And finally, I wanted to talk about this technique, new technique of sequencing that allows to take long reads of DNA or RNA. This is based on nanopore, in which the DNA strands is sliding. And it's possible to make a read of the sequence by analyzing the current signal with deep learning techniques also. This is very interesting because it allows to take a look at states modification of the DNA, particular methylation, that is very important to determine how the DNA is read. These epigenetic marks, there are modifications of the DNA and proteins that are involved, are very important, for example, in determining cell fate. For example, what is different between an epidermal cell and a neuron that share the same DNA actually, but differ in these epigenetic marks. This is our team, or a part of it. This is our director, Stefano Cozzini, and we are quite young and growing team. And I suggest you This, by the way, is the infrastructure that we have, our genomics and epigenomic laboratory and the data center that is in charge of analyzing all this data. And please feel free to contact us if you want to know more about our activities. And I warmly thank you for the attention.